Today I'll teach you about the difference between cyclic phosphorylation and non-cyclic phosphorylation, and how the ATP is formed in cyclic phosphorylation. And next to this is the pseudocyclic photophosphorylation. And next to this is photooxidation of water molecule, and about red drop effect and Emerson enhancement effect, and about pigment structure, formula, their distribution and absorption maxima. And lastly, I'll teach you the discovery and history of photosynthesis. Now we come to the point cyclic and non-cyclic phosphorylation. The basic difference between cyclic and non-cyclic phosphorylation are listed here. The first difference is that in cyclic phosphorylation only photosystem 1 participates, whereas in non-cyclic phosphorylation both PS1 and photosystem 2 participates. The second difference is that in cyclic phosphorylation the system works like a closed loop, and the electrons emitted or ejected from the reaction center come back to its original place, whereas in non-cyclic phosphorylation it requires the free electron donor, and here the H2O, that is the water molecule, acts as the source of free electron, and here NADP is the final electron acceptor, and the electrons emitted or ejected from the reaction center does not come back to its original place, that is at PS2. And the third difference between cyclic phosphorylation and non-cyclic phosphorylation is that in cyclic phosphorylation, the reducing power NADP is not reduced, so that the rate of CO2 assimilation is reduced. Whereas in non-cyclic phosphorylation, the reducing power NADP is reduced to NADPS2, which is utilized in CO2 assimilation. The next difference is that in cyclic phosphorylation, oxygen evolution does not take place. So in here at PS1, the oxygen evolution does not take place. It means there is no photooxidation of water here. Whereas in non-cyclic phosphorylation, oxygen evolution takes place because photooxidation of water takes place at photosystem 2. And in cyclic phosphorylation, the path of electron or the transfer of electron takes place from photosystem 1 to X, that is the unknown complex, and from X to FRS, followed by FD, and followed by cytochrome B6, and then to plastocyanin, and then from plastocyanin to cytochrome C, and then to plastocyanin, and lastly to photosystem 1. Whereas in non-cyclic phosphorylation, the transfer of electron is from photosystems 2 to quinine, from quinine to cytochrome B6 to plastocyanin to cytochrome F, and from cytochrome F to plastocyanin, and then to PS1, that is the pigment system 1, to X, that is unknown complex, from X to FRS, and then FD ferredoxin, and then from FD reductase, that is ferredoxin reductase, and from ferredoxin reductase to NADP, and lastly NADPS2 form, that is the reducing power. And the next figure is that how the ATP is formed during cyclic phosphorylation. This picture shows how the ATP is formed during cyclic phosphorylation. This is the photosystem 1, and this is the here in the antenna molecule or the pigments are present, and this is the reaction center. And from photosystem 1, it absorbs the pigments present in photosystem, absorbs light energy, and then transfer or migrate this light energy to reaction center, and after getting light energy this system becomes excited and it releases its electron, and this electron goes to this here, and from this excited system it comes back to again photosystem via different electron carriers. And this diagram shows the electron transport chain, and these are the carriers. And when the electron passes from cytochrome B6 to cytochrome F, it releases some of its energy, which is utilized by ADP and inorganic phosphate for the formation of ATP. And this type of ATP formation is known as cyclic phosphorylation, and this scale is the redox potential of different electron transport carriers, and which is present in the electron hole form. And the next slide is, or sorry, the next topic is that pseudocyclic phosphorylation. This is another type of photophosphorylation known as pseudocyclic photophosphorylation. Here, the one molecule of water molecule in presence of illuminated chloroplast in presence of FMN forms ATP, and this FMN is present is reoxidizable, and when it combines with water, the water molecule breaks down, and FMN oxidized FMN is get reduced to FMNS2, and then it combines with oxygen to form again this form water molecule, and this becomes oxidized. This is the total reaction takes place here, and this process of pseudocyclic phosphorylation was first demonstrated by Arnon and his co-workers in 1954. And they showed that this is the another kind of photophosphorylation, because this process also takes place in the presence of light. They observed that even if chloroplast molecule are illuminated, they can produce ATP from ADP and inorganic phosphate in presence of FMN and vitamin K and oxygen. And this process is very simple and require no net chemical change for the formation of ATP and water. Arnon called this is as oxygen dependent FMN catalyzed photophosphorylation or pseudocyclic photophosphorylation, which involves reduction of FMN with the production of oxygen. FMN, that is flavin mononucleotide, is reoxidizable hydrogen acceptor with the effect that the reduced FMN is reoxidized by O2. Thus, the process can continue repeatedly to produce ATP. Arnon further demonstrated that the source of both hydrogen and electron from the reduction of FMN and of oxygen for its oxidation is the water molecule. The FMN, that is the flavin mononucleotide, rejoins the water molecule. Since the process can be self-repeated, it appears that a single molecule of water should be sufficient to operate pseudocyclic photophosphorylation to meet the requirement of ATP. And this is the reaction which shows the whole process of pseudocyclic photophosphorylation. This is FMN. This is flavin mononucleotide. It is in oxidized form. When it combines with water molecule, it becomes in presence of illuminated chloroplast, it becomes reduced and oxygen is formed. During this process, ADP and inorganic phosphate is converted into ATP, and this process is a reversible process, and this reduced FMNS2 combines with oxygen, and this water molecule again forms, and FMN becomes oxidized. So this shows that a single molecule of water breaks down, and it breaks down and forms hydrogen and oxygen, and this hydrogen is used by FMN to make it in reduced form, and oxygen is liberated, and this oxygen again combines with this reduced FMNS2 and forms oxidized FMN and water molecule. And in this process, when this come back, this FMNS2 is oxidized, and when this process is oxidized, this ADP and inorganic phosphate forms ATP molecule. So the ATP molecule is continuously synthesized, and a single water molecule breaks down and again reduced to water molecule. Now we come to the photooxidation of water. As we know, the name photooxidation, photo means light, oxidation means breakdown. That means in presence of light, the breakdown of water molecule. The process of photooxidation of water takes place at photosystem 2 of light reaction. And this photooxidation of water is the first photochemical reaction in photosynthesis. And this photooxidation of water and evolution of molecule takes place during this photochemical reaction. It is mainly associated with pigment system 2. The photooxidation means the breakdown of water molecule or the photolysis of water molecule takes place at pigment system 2. The light energy is first harvested by antenna complex PS2 and then transferred to its reaction center P680. It means the light energy which is absorbed first, harvested by antenna complex of PS2, and then it is transferred to the reaction center of P680. And after getting this light energy, the P680 gets excited, and this excited P680, the life it has very short life, that is about 5 into 10 to the power minus 9 second, is immediately transfer its electron to adjacent primary electron acceptor. And the transfer of electron from P680 creates a gap. It means when P680 releases an electron, there creates a gap which is filled by electron produced by the photooxidation of water molecule. And this water molecule are oxidized into hydrogen ion and hydroxyl ion uh, as follows. Firstly, this water molecule in presence of light and chlorophyll gets break down into hydrogen and hydroxyl, and this hydrogen, when releases electron or emitted an electron, it becomes hydrogen ion, and this hydroxyl and when gets electron, it becomes hydroxyl ion. And here and this hydrogen ion and hydroxyl ion is formed, and the transfer of electron to P680 takes place via an unknown compound Z. The hydroxyl molecule reunite to form water and evolve molecular oxygen. And this step depends on some enzyme and requires manganese ion, chlorine ion, and calcium ions. And the protons are carried by some carriers, that is plastoquinone. So the transfer of electron from P680 takes place via an unknown compound, that is Z, and which is shown in a light in electron transport chain. And this hydroxyl ion which is formed is reunite with this hydrogen ion to form reunite to form water and evolve molecular oxygen. And this step, and that means the oxidation of water molecule, needs some manganese ion, chlorine ion, and calcium ions, and the protons which is released during this photooxidation of water molecule is carried by some electron carriers, that is plastoquinone. According to Koch et al. In 1970, that is Koch et al. In 1917, shows that water oxidizing enzyme complex exists in 5 states, that is S0, S1, S2, S3, and S4. This complex is called S state, and this all 5 states combine together to form S state, and the removal of oxygen from two molecule of water is as follows. That is, water, when this S state is in S0 form, the water molecule combines with this, and this S0 after receiving one photon of light becomes transformed into S1 state, and this S1 state again converted into S2 state by getting one photon of light, and this S2 is again changed into S3 state after getting one photon of light, and from S3 to S4 again from one photon of light. So here we see that S0 is transformed into S4 state by getting 4 photon of light. And here simultaneously this water molecule combines with S0 state and changed into hydrogen ion, and oxygen is evolved. This is all about the photooxidation of water molecule. Now to the red drop effect and Emerson enhancement effect. uh, uh, red drop and Emerson enhancement effect. The red drop, this is an important phenomenon, and when photos this is an important phenomenon. इसको काट दो पहले. Robert Emerson and Lewis 1943, while determining the quantum yield of photosynthesis in chlorella by using monochromatic light of different wavelengths, noticed a sharp decrease in quantum yield at wavelength greater than 680 mu m. Because this decrease in the quantum yield took place in the red part of the spectrum, this phenomenon was called as red drop effect. जैसा कि आप जानते हैं कि रॉबर्ट इमर्सन एंड लेविस 1943 में जब उन्होंने क्वांटम इल्ड फोटोसिंथेसिस के क्वांटम इल्ड की स्टडी की थी क्लोरेला प्लांट पे, तो उन्होंने सबसे पहले क्या किया कि मोनोक्रोमेटिक लाइट लिया. मोनोक्रोमेटिक लाइट मीन्स लाइट हैविंग सिंगल कलर. इट मीन्स कि डिफरेंट वेवलेंथ्स की लाइट को लिया और उन्होंने उस पर उन्होंने फोटोसिंथेटिक इल्ड को मेज़र किया. फोटोसिंथेटिक इल्ड जो मेज़र किया जाए दैट इज़ इन द फॉर्म ऑफ ऑक्सीजन इवोल्यूशन. तो उन्होंने देखा कि डिफरेंट वेवलेंथ्स को अलग अलग लेके उन्होंने क्लोरल प्लांट पे अप्लाई किया, सप्लीमेंट किया, उसको ट्रीट किया. उन्होंने देखा कि जब डिफरेंट वेवलेंथ की लाइट को लेते हैं तो एक रेंज पे, क्योंकि हम जो विजुअल रेंज में जो पार है हमारा फोटोसिंथेटिक एक्टिव रेडिएशन, इसमें डिफरेंट वेवलेंथ की लाइट है और डिफरेंट कलर की लाइट है. तो वो जब 680 म्यू एम के करीबन पहुँचे, उससे ज़्यादा वेवलेंथ की लाइट पे देने लगे क्लोरेला प्लांट को, तो उसमें क्या है फोटोसिंथेटिक इवोल्यूशन जो है अचानक से ड्रॉप कर गया. ड्रॉप करने से के बाद उन्होंने देखा कि स्पेक्ट्रम में वो वेवलेंथ की लाइट जो है वो रेड लाइट के रेंज में आती है. इसलिए चूँकि रेड लाइट में फोटोसिंथेटिक इल्ड जो है वो अचानक से ड्रॉप कर गई, और यानी कि ऑक्सीजन इवोल्यूशन ड्रॉप कर गया, इसलिए ऐसे इस इफेक्ट को हम कहते हैं रेड ड्रॉप इफेक्ट. एंड दिस ग्राफ ऑल्सो शोज कि जब ये वेवलेंथ है और ये आपका ऑक्सीजन क्वांटम इल्ड है इन द फॉर्म ऑफ ऑक्सीजन इवोल्यूशन. रेड ड्रॉप मीन्स 660 के बाद 680 के बाद आपका ऑक्सीजन इवोल्यूशन जो है वो 0.1 है, जो कि अचानक से गिर के 720 तक आके एकदम 0.02 पे पहुंच जाता है. सो इस तरीके से जो ड्रॉप हो रहा है फोटो से ये क्वांटम इल्ड में, एंड दिस इज नोन एज द रेड ड्रॉप इफेक्ट. लेकिन इसके बाद एमर्सन एंड हिज को वर्कर्स आए, उनका पूरा ग्रुप आया, जो कि बाद में उन्होंने एक्सपेरिमेंट किया. उन्होंने कहा कि इनएफिशिएंट जो फार रेड लाइट है, फार रेड लाइट मीन्स दैट इज इंफ्रारेड लाइट, जिनकी वेवलेंथ जो हमेशा 7000 आर्मस्ट्रॉन्ग से ऊपर की होती है, एंड दे आर इनएफिशिएंट इन एज फार एज फोटोसिंथेसिस इज कंसर्न. तो जब इनएफिशिएंट लाइट फार रेड लाइट को क्लोरेला में देते हैं, जो कि 680 म्यूएम से ऊपर की होती है, इसको वो ज़्यादा इफिशिएंट बना देते हैं यदि इसी लाइट के साथ हम किसी शॉर्टर वेवलेंथ की लाइट को मेज़र करें. तो जब ये शॉर्टर वेवलेंथ की लाइट और ये इंफ्रारेड लाइट जो है दोनों एक साथ दी जाती है, तो जो क्वांटम इल्ड जो होता है वो हमेशा जो है दैट मीन्स क्वांटम इल्ड ऑक्सीजन इवोल्यूशन जो है वो हमेशा बढ़ जाता है. और वो जो होता है वो हमेशा किससे ग्रेटर होगा, जो कि डिफरेंट वेव ये फारेड लाइट में जितना ऑक्सीजन इवोल्यूशन हुआ क्वांटम इल्ड हुई, और जो उससे जो शॉर्टर वेवलेंथ की लाइट में जो क्वांटम इल्ड हुई, दोनों का सम कर दिया जाए उससे ज़्यादा जो होता है जब इन दोनों लाइट को एक साथ ये फारेड लाइट को इसको सप्लीमेंट करते हैं शॉर्टर वेवलेंथ की लाइट से. एंड दिस तरीके के इफेक्ट को जो होता है वो इसको हम लोग कहते हैं, चूँकि एनहांसमेंट हो जाता है फोटोसिंथेसिस में या क्वांटम इल्ड में, इसको हम और इसको प्रूफ किया था एमर्सन ने, इसलिए इस इफेक्ट को हम लोग कहते हैं एमर्सन एनहांसमेंट इफेक्ट. यहाँ आप ग्राफ में देख रहे हैं कि ये जो है ये वेवलेंथ शॉर्टेड वेवलेंथ की लाइट है 660 ये है, और इसको जो है हम ये सप्लीमेंट करते हैं सप्लीमेंट्री लाइट के साथ वो करते हैं, सॉरी ये जो ये हायर वेवलेंथ की लाइट है, जब इसको हम सप्लीमेंट करते हैं ये शॉर्टर वेवलेंथ की लाइट से, तो यहाँ जो है फोटोसिंथेटिक इवोल्यूशन जो है फोटोसिंथेटिक ये ऑक्सीजन इवोल्यूशन जो है वो हमेशा ऊपर जा रहा है, यानी ये फोटोसिंथेसिस के रेट को बढ़ा रहा है, एंड दिस इज क्वांटम इल्ड इसमें बढ़ रहा है, एंड दिस इफेक्ट इज नोन एज एमर्सन एनहांसमेंट इफेक्ट. Next to this, we will discuss about the structure, formula, and this is the distribution and absorption maxima of photosynthetic pigment. This picture is about the chlorophyll structure. This shows the chlorophyll structure, and this table shows about the pigment, about the pigment, their formula, their distribution, and absorption maxima. This is the basic structure of chlorophyll molecule, and this is here is the tetrapyrrole ring, ring 1, ring 2, ring 3, and ring 4. And this tetrapyrrole is centrally here the centrally placed magnesium ion, centrally placed magnesium ion. And in chlorophyll A, the ring 2 has methyl group, as whereas in chlorophyll B, this methyl group is replaced by aldehyde group. This is the basic difference between chlorophyll A and chlorophyll B. And this tetrapyrrole ring is combined with a phytol chain. This is the phytol chain. This is the structure of chlorophyll molecule A and B. And now we come to the chlorophyll C. This chlorophyll A is having the formula of C 55H72O 5 and 4 Mg. It is found in all green plants, and the absorption maxima is 436 for 670 680. And this chlorophyll B, and this is the basic structure C 55H70O5 and 4 Mg. It is found in all green plants, and this except red, brown, and diatoms. Blue-green algae, and absorption maxima is 453, 480, and 650. And this is about the chlorophyll C, D, and chlorophyll A. Bacterochlorophyll A, bacterochlorophyll B, and bacterioviridin. And this next to this is the carotenoids. Carotenoids having carotene and xanthophylls. And then these carotenoids are present in red, green alga, and all green plants. In all green plants, green bacteria, and the xanthophylls present in red, green algae, all plants, green leaves in brown algae. And then phycobilins are present in blue-green algae and red algae. And the absorption maxima of carotenoids, that is carotene, is 450, 470. And then about xanthophylls, the absorption maxima is 425, 475, 425, 450, and 475. And the absorption maxima of BCA is 618, and red alga is 490 and 576. This is all about the pigments structure and their formula, distribution, and absorption maxima. And now the next to this I will discuss about the history and discovery of photosynthesis processes. This table shows the discovery of different scientists in the process of photosynthesis. For example, the one Helmont concluded by his experiment that dry weight of plant increases due to water absorbed by them and not due to soil. And next to this is that this Jan Ingenhorst concluded by his experiment that green plants purify noxious air during the day in presence of light, and he proposed the following equation: CO2 plus H2O in presence of light and green plants, oxygen is evolved, and plus organic matter. And then to this Sachs shows that Sachs demonstrated that the first visible product of photosynthesis is stars. He also showed that chlorophyll is confined to the chloroplast. Engelmann showed that chloroplast was the seat of photosynthesis in the cell, and he discovered the role of different wavelengths of light on photosynthesis and plotted the action spectrum. Blackman in 1905 discovered two steps, that is light and dark reaction in photosynthesis. He further proposed the law of limiting factor, and Warburg shows that performed flashing light experiment using green alga chlorella as a suitable material for the study of photosynthesis. Van Neel in 1931 demonstrated that some bacteria use H2S instead of water molecule in the presence of photosynthesis. Arnold carried out the flashing light experiment at different temperatures and showed the existence of light and dark reactions. Whereas this Melvin and Calvin in 1954 used chlorella as an experimental material, discovered the first double product of photosynthesis, that is 3-phosphoglyceric acid, traced the path of carbon using 14 CO2 in photosynthesis, and Calvin cycle in C3 cycle, and awarded the Nobel Prize in 1961. Arnold, Allen, and Watley used 14 CO2 and demonstrated fixation of CO2 by isolated chloroplast. Hill and Bendal proposed Z scheme and suggested that two photosystems operated in series. Anand in 1960 One, discovered photophosphorylation. Hatch and Slack trace CO4 C4 cycle for CO2 fixation in certain tropical grasses. Hatch and Slack cycle. And next to this is all about the history and discovery of photosynthesis. And next class I will teach you about the C3 plants, C4 plants, the difference between C3 plants and C4 plants, about Krebs anatomy, and C4 cycle, C3 cycle. C3 cycle, that means the Calvin cycle. C4 cycle, that is the Hatch-Slack cycle. And about cam plants and photorespiration and physiological and ecological consideration uh, considerations. And that all these topics I will discuss in the next class. Thank you.